Okay, hi. So I'm going to do a quick little overview of Lab 12 and what we're up to. So it's not ideal that you need to do this at home. I would much rather you do it in lab, but this is where we're at. So this is about different structures of the plant, such as roots and stems. So roots are to anchor the plant and also play a role of absorbing water and minerals. So let's look at the different parts of a root. So we have vascular tissue, aka xylem and phloem. Xylem is what's going to conduct water, and phloem is what's going to conduct the products of photosynthesis. We also have a really important tissue, a layer of cells known as the endodermis. So the endodermis is this layer right here. The endodermis surrounds the vascular tissue, and we call this cylinder of vascular tissue the stele, S-T-E-L-E. So this endodermis helps regulate the materials that can enter the vascular tissue. Because once something gets in our vascular tissue, it can get throughout the entire plant. So you don't want just any old thing that gets absorbed from the soil to be able to enter the vascular tissue, like a toxin or something, you know. So this allows it to have some selectivity and not let bad things in. So this endodermis has a waxy layer called the Casparian strip, which you can see right here in the purple. And what this Casparian strip does is it forces water and anything that is in that water to have to go through the cytoplasm of the cell rather than just get sucked through the cell walls. Because these cell walls, the water can just kind of make its way along what we call the apoplastic pathway, just not going through the actual cell, kind of making its way through the cell walls. What's called the symplastic pathway means you actually have to go through the cell, which means you have to make it through the semi-permeable membrane, which has the ability to be selective about what can and cannot pass. So this waxy Casparian strip basically is a stopgap to say, no, you have to go through the cell at this point so that we can control what goes in and out, what actually makes it to the vascular tissue. So that's important. We also have what we call the cortex. So the cortex is ground tissue that is made of those parenchyma cells. So if you haven't listened to the plant tissues lecture yet, the parenchyma cells take on a number of different roles, one of which is storage, and that is oftentimes their role in the root. And then usually there's a thin cuticle on the outer layer of cells known as the epidermis. We want the cuticle here to be nice and thin because we need to absorb water. Oftentimes we also have root hairs that increase the surface area over which the roots can absorb water. So a big thing here in this lab is distinguishing between a group of plants we call the monocots and a group of plants we call the eudicots. The monocots get their name because when they are in their seed form, they have one embryonic seed leaf that we call a cotyledon. Whereas the eudicots have two embryonic seed leaves, and that's a really awful drawing, but it's hard to draw with this pen. That's where they get their names, and they differ in the arrangement of their vascular tissue in their stems, in their roots, and their leaves. And that's the thrust of what we're looking at in this lab. Okay, so let's look at a monocot first. So this is our monocot. And what we have going on here is that the xylem and phloem are found in alternating patches. So here are our xylem. The xylem, by the way, is almost always larger in diameter than the phloem. And then here's our phloem. And as you can see, they're in alternating patches. Xylem, phloem, xylem, phloem, xylem, phloem, etc. All the way around in a circle. So what we have in the middle, this patch of parenchyma cells in the middle, we call this the pith, located inside this vascular tissue. So we then have our endodermis, and then we have this area of parenchyma we call the cortex. So typically if our ground tissue is inside the vascular tissue, we call it pith. If it's outside of the vascular tissue, we call it cortex. In a eudicot, we have a very different situation. We don't have pith. what we have is a dense concentration of vascular tissue in the very center. And so what we have are our xylem is in this sort of cross shape. Sometimes we call this the xylem arms. And then we've got patches of phloem in between them. So we have this very concentrated vascular cylinder, and then we have a very thick cortex outside of it. So here's just a side-by-side of the two. So you're going to be looking at a Monocot root slide and drawing it and labeling the epidermis, endodermis, cortex, pith, xylem, and phloem. And let's go back to the endodermis here and remind you that the endodermis, so here's the endodermis just outside of the vascular tissue in the Eudicot root, and here's the endodermis in the Monocot root. So it's just outside the vascular tissue. And you're also going to look at a Eudicot root slide. Now, here is the thing that is a little bit annoying with this, is that obviously you don't have a microscope at home. So what I have is a PDF labeled Lab 12 Images for Drawings. So we have our Monocot root. And you gotta do a bit of scrolling, and there's a Eudicot root. So you're going to use these images. And if you scroll down further, we have the rest of the images on there. We have our Monocot stem, our herbaceous Eudicot stem, our woody Eudicot stem, our Monocot leaf, and our Eudicot leaf. So all of these things we're doing drawings of. So that's the document you're using for that. Oops, I'm ahead of myself here. Okay, so stems have the role of supporting plant structures. They also transport water, nutrients, and sugars. They've got vascular tissue running up through them. They do not, however, have an endodermis. They don't need one. Everything is already in the vascular tissue, right? You're not having things absorbed through the stem, so you don't have to worry about it. The xylem and phloem, rather than being in a central stele like we see in the root, are in what we call vascular bundles. So here is an example of a vascular bundle, basically a little bundle of xylem or phloem. And we can imagine this little bundle, this is in cross-section, so it would be running the entire length of the stem. In Monocot stems, vascular bundles are scattered throughout the stem, and in Eudicot stems, vascular bundles are arranged in a ring with the pith in the center. And we usually have a small amount of cortex. In a Monocot stem, we really don't have a differentiation between pith and cortex. We just call it ground tissue because our vascular bundles are scattered within it. So do I have an up close? I don't have an up close vascular bundle here. So let me get one. Okay, I wanted to look at that a little closer. So slideshow, current slide. Okay. Let's look at what's going on here. So phloem, I always think the Monocot vascular bundle looks like a little face, and the forehead is the phloem. Okay, so there's our phloem. And then we have our much larger xylem, and then we have some very thick-walled cells called sclerenchyma around it, helping be protective. In our herbaceous Eudicot stem, so here are our xylem cells. Xylems, again, usually larger in diameter. Here's our phloem cell. And then we typically have a cap of thick-walled sclerenchyma cells on the top as well that help make them stronger. So, xylem, phloem, sclerenchyma. Xylem is usually the bigger, oh, I can't do it there, but you get the idea. Okay, so another thing I need to talk about is primary versus secondary growth. Because some eudicots are capable of secondary growth. Not all of them, and no monocots can do secondary growth, just eudicots. This is why they have vascular bundles in a circle. So primary growth means growth in length. So if you're growing up or growing down, that's primary growth. Secondary growth is growth in girth. You're getting thicker and thicker. Primary growth happens at regions called apical meristems. These are at the tips of roots or the tips of shoots. You can review my gosh, I can't talk today. I did a PowerPoint, I forget what chapter in your book. Chapter 33 covers apical and lateral meristems. So let's talk about our lateral meristems for a second, because we see these in Eudicot stems, because we're gonna get growth and girth. They're also in Eudicot roots. So in addition to an apical meristem, we have what's called our lateral meristems. And there are two important ones in a Eudicot stem. One is going to be found if you look at the vascular bundle, there's a line there between the xylem and the phloem, and this line is called the vascular cambium. This vascular cambium is going to put on secondary xylem to the inside and secondary phloem to the outside. So the first xylem and phloem that was made through primary growth we call our primary xylem and our primary phloem. The layers of xylem and phloem we keep putting on through secondary growth we call secondary xylem and secondary phloem. So remember, xylem is larger in diameter, so it's going to make up the bulk of the woody tree. And so what we think of as wood is actually this secondary xylem, layers and layers of this secondary xylem. So let's look at this actual cross-section through a woody stem. So here we have our annual growth rings, and I know we've all counted annual growth rings looking at a cross-section of a tree, but what you're actually looking at is our secondary xylem. And the reason you can distinguish these clear growth rings is because this secondary xylem is put down throughout the year. And early in the year, we have what's called early wood. In the early wood, which is earlier in the spring, the diameter of the xylem is much larger because there's more water available, and so it appears lighter. Later in the season, in the summer where water is more scarce, the secondary xylem is much more narrow in diameter, so it appears darker. And this layering of light and dark secondary xylem make up that annual growth ring. So we could look, and we start here, and we could say there's 1, 2, 3 annual rings, so this is a 3-year-old stem. Okay, so you want to make sure you can identify the vascular cambium, which would be right here, and also another lateral meristem called the cork cambium. The cork cambium puts on these cork cells to the outside only. These are waterproof, very tough cells. We call it the periderm. It's what's going to replace the epidermis in woody eudicots. So the periderm, along with the secondary phloem here, we collectively call the bark of the tree. Oh, look, it's all right here. Sorry. So, two types of lateral meristems, vascular and cork cambium. Okay, I think I said all this, but, we also have it up there. Okay, so you're going to look at stem slides. And, of course, you're not looking at slides. You're just going to be looking at the pictures I present. So here are your labels: epidermis, vascular bundle, pith, if present. For the woody slide, periderm, cork cambium, vascular cambium, secondary xylem, secondary phloem. So finally, looking at leaves. This is the primary organ for photosynthesis. Contains both a upper and a lower epidermis. Usually, leaves are very thin and flat because they want to intercept as much light as possible. They're covered by a waxy cuticle that helps keep excess water from being lost. They have pores called stomata that regulate gas exchange and transpiration. So this is a magnification of the stomata. So these little pores will allow O2 out and CO2 in for photosynthesis, and also allow water vapor out. So monocot leaves have stomata on both layers of their epidermis. Most eudicots have stomata on the lower epidermis only. So let's look at a leaf's interior. This is an example of a eudicot leaf. So most of a leaf's interior is made of what are called mesophyll cells. Mesophyll cells have large number of chloroplasts, so a mesophyll cell is our main cell for photosynthesis. There are two main types of mesophyll cells in a eudicot leaf. These barrel-shaped cells are called the palisade mesophyll, and they're usually aggregated very close excuse me, to the epidermis. And then further down we have what are called the spongy mesophyll, which are more loosely arranged so that we can allow for gas exchange. And then the stomata are on the bottom here. In monocots, we don't have this differentiation between spongy and palisade mesophyll. The mesophyll is more uniform. And we just call it, typically we just call it mesophyll. Okay, so here's an actual cross-section of leaves. So most monocot leaves have parallel venation. So think of a grass or the leaf of a corn or something like that, where the veins are literally parallel. So not surprisingly, when you look at this in cross-section, what do you see? You see several vascular bundles that are roughly of the same size. Now, usually there's a central vein that's a little bit bigger than the others. That's pretty typical. So you want to pick out the xylem and the phloem and the mesophyll and as well as stomata if visible in your drawing. And I know that I've just given you the image, so you don't have much to pick from. You can't sort of zoom around, so just draw what you can. Okay, so looking at our eudicot leaf, so we have our clear, here's our palisade mesophyll, and then further down is our spongy mesophyll. And because of the venation pattern, we end up with one really big vascular bundle in the center here. And then smaller, you might see smaller vascular bundles on the side. Here we have a vascular bundle, but it's kind of in long section, right? Because it's branched, so sometimes some will be running a different orientation than that central main vascular bundle. Okay, so again, we have images for you to look at, label epidermis, mesophyll, vascular bundle, and stomata if visible. And then finally, we're going to talk about differences between monocot and eudicot flowers. So monocot flowers typically have parts in 3s, whereas eudicots have parts in 4s or 5s. So it might have, for example, 5 petals, 5 stamens, 5 pistils. Whereas a monocot might have 3 petals or 6 petals, 6 stamens, and a pistil that has 3 chambers. So monocots, 3s or multiples thereof, dicots, 4s and 5s or multiples thereof. Now in our actual lab, we have plants on display, and you are going to determine if they're monocots or eudicots and give 2 reasons that support your identification. Now obviously, we're not in lab, so instead, let's see if I can figure out where I put it. I have images again. So for example, number 1, this is a monocot. And I would be able to tell you it's a monocot because it's got parallel venation. In this case, because all I'm showing you is the leaf, you can't tell me anything else. So you're just going to write number 1, monocot, parallel venation in parentheses. Number 2, this is a eudicot. How do I know? I actually have 2 things that can support my identification this time. First, flower parts 1, 2, 3, 4, 5. Flower parts in 5s. Also, netted venation. So I'm only going to give you those 2. The rest you got to figure out yourself. So this document is called monocot or eudicot, and it's a PDF. And then, at long last, the final thing I want you guys to be aware of is another PDF that I have labeled, titled Lab 12 Models and Images. Because here's the thing. We have a lot of models in lab that could turn up on the lab exam. But again, because of I'm glad we have a holiday, don't get me wrong. But we just don't have time to get this lab in. So I have taken pictures of the models that are in lab. So this is an example of one of the models. So you should be able to identify things like the xylem, the phloem, all this stuff off a model. So take some time. There's nothing you have to actually write down or anything like that here. But I just want you to look this over and be familiar with it, okay? Like this leaf, you should know the spongy mesophyll, the stomata, the vascular bundle, which is kind of truncated or cut off in this case, the palisade mesophyll. You should know all those features. Okay, so go ahead and look over that as well. That's important because it could be on your lab practical. Okay, I think that's all I have. Please let me know if you have questions. I know I went over this a little fast. Yeah, so you're just going to be filling this out at home and then bringing it to lab. Also, I put a review PowerPoint for the lab practical, and that's the other thing we're doing. So please let me know if you have any questions.